Hello? Can you hear me? Good. Okay, I think we should probably make a start. Welcome to the final session of this CCP4 Study Weekend. We have 4 talks. I'd just like to quickly request the speakers to try and keep to time as people have taxis and buses to catch at the end of the session. So without further ado, we'll start with Giovanna Scappin from Merck, who's going to give us a historical perspective on molecular replacement, I believe.
Can you hear me? Okay, good. First of all, I would like to thank the Pietro for inviting me here to give a talk. And when Pietro asked me to give a presentation on molecular replacement, and I quote, he said about the history of molecular replacement, the development of the method, and what we, crystallographic community, require in a model, what we want from a model to the quality of the model to be in order to use it as a molecular replacement probe. I was very excited because it's always nice to be invited to a meeting. I was also a little bit perplexed, and I said, why me? But then I decided, okay, let's do it. It's going to be fun. And it was fun, although not completely fun the way I initially intended to. So in my mind, molecular replacement should be a very straightforward. To me, is the easy way to go, the molecular replacement. Much easier than doing heavy atom soaks or selenium metal, all that kind of stuff. And all you need is have a good data set, a good model, and you can get a solution relatively easy. Sometimes, many times, 95% of the times. But then there is always that 5% of the times that things don't work. And then you're stuck because you don't really know what to do and to solve this little problem over here. And I was thinking about it, and I was thinking about how molecular replacement techniques have changed over the years since 1980 till today. And at that point, I realized that what my "yes, Pietro, I will do it" actually meant was that I will be here standing in front of you, talking to you about the history of molecular replacement and the development of the methods, and I will talking to the very people that made this happen. And it's like me standing in front of the Yankees, telling them about the history of baseball, and how great is baseball, and how wonderful, and this is what you should do to play baseball. Didn't sound very nice to me, and so I panicked for a couple of weeks. And then basically I said, okay, but one thing I am, and I am a user. And I've been a user for many, many years, many more than I care to remember. And I'm a user in an industrial setting, which is a little bit different than the usual academic setting. We do things really fast. We are asked to solve the problems one after the other, after the other, after the other, without really dwelling too much in why things work or don't work. And so what I decided to do is to give you my perspective, my personal view of the molecular replacement evolution during the years, using some of the examples that I've been faced with. And if you think that you will be seeing a lot of things that you saw already, you're right. Because, first of all, this is a little bit of an overview. Second of all, because no matter how unique our problems, how unique we think our problems are, they are never unique. There is always somebody else there that has exactly our same problem, and that solve the problem in a different way. And that's maybe it's good that we go to meetings, because we get to interact and we got to figure out that we are really not alone in this world. So the concept of molecular replacement was started in the early 60s, and the term molecular replacement was actually the title of a book that Rossmann and Bloch assembled with a few initial papers on the topics and their reviews on the papers. And since then, it has really gone a very long way. And this is just a distribution of the number of structures that have been deposited in the PDB and that have been solved by molecular replacement. And there is clearly an exponential trend. And it becomes even more impressive if we compare the number of structures that have been solved by molecular replacement to the total number of structures. Basically, over 60% of the structure have been solved by molecular replacement, and if you look in the last 2 years only, is about more than 70%. So molecular replacement is definitely the method of choice. Okay, granted, a lot of these things are the same thing. So there are 2,300 structure of the same enzyme bound to different cofactors. But still, it's a very impressive growth. And of course, what made this possible has been a very drastic evolution in the programs associated with the fact that we have a much better computer hardware right now. We can do now things that 20 years ago were not even thinkable, not because we didn't know how to do it, or we did not have the idea how to do it, but because there was no hardware capable of sustaining these ideas. And also, there have been, this is almost dead, major evolution in the models, not just in the type of models that we have, but also in the number of models that we can pick from, we can choose from. So I'm going to go and just talk a little bit about this, my history in the different programs that I used during my career. Okay, so this is a distribution, again, taken from the PDB. So if you, in the search field of the PDB, you type molecular replacement and Amore, you get the number of structures that have been solved using Amore throughout the years. And then you can slice it and do whatever you want with those numbers. And so as we can see here, there are basically 4 major programs that have been used to solve the large majority of the structures in the protein database. But we shouldn't forget what I call the others, and which are the very programs that were developed at the very beginning. And probably most of you don't even know about the program or never heard about them. But those are the basis, or most of them are the basis, upon which every other program has evolved. My personal big breakthrough, a program that I loved at the beginning, was Explore and CNS. And not just Explore CNS, what I usually loved was the Patterson correlation coefficient that was implemented in Explore and CNS. And that was, I think, the first idea that when we do a molecular replacement, we don't take a ball and we move it from here to here, but we take a molecule that is flexible, that has things that move, that have domains. And sometimes, or at least back then, it was really hard to deal with this flexibility. And what the Patterson correlation coefficient, Patterson correlation refinement gave us basically a way to adjust these different domain positions. This is a very quick summary of this paper, in which basically that brought the method to popularity, and is the application when you're trying to solve an FAB molecule. An FAB molecule is normally formed by 2 domains, a constant and a variable domain, and they are joined by a linker. And so the relative position of the 2 domains is always different in a different FAB. So if you have your unknown FAP and a search model, the relative angle between the 2 domains is probably not the same. So you can get a series of rotations that tend to match the individual domains, but they are very low level, and it's really hard to define between all these rotations the one that will actually give us a correct translation. What the PC refinement does is to adjust the relative position of the 2 domains and generates a model that eventually will give us a perfect translation. I used this program many, many times. It gave me some great success, some great failures, like everything. One thing that CNS or Explore had was slow, very, very, very slow. And I got to a point in which I needed something to be much faster, and that's when I moved to using Amore. Amore, it was complete package. It was way faster than Explore. That's why I liked it a lot. And it also was much more flexible, or at least I felt that it was much more flexible, and allowed me to use different input models, but also different input space as a search space. And what I'm going to give you now is an example, something that I've done many years ago, and I was very proud because I was the way I solved the structure was a kind of innovative back then. Nowadays, of course, it's not. But it also shows the flexibility that this Amore gave to us, to postdoctoral crystallographers. So, pane, amore, fantasia, bread, amore, and a lot of imagination, because that's what I had to use back then. The system was a protein, a bacterial protein that we were looking at as a target for antibacterial. It was known that it was the biological form was a tetramer. In my tetramer, the protein binds a cofactor and a substrate. The binary complex with cofactor only crystallized with one molecule per asymmetric unit, and a tetramer was generated by crystallographic symmetry. About 6 months down the road, I managed to get crystals of the ternary complex, which was what we wanted, because we wanted to be able to devise the mechanism of the enzyme and everything else. Not as good as these guys, lower resolution. There were 4 molecules, hopefully a tetramer per asymmetric unit. And so I said, okay, easy, a tetramer, I'll take the tetramer, I'll do this molecular replacement. No way. So I thought, okay. So I tried with the monomer, I tried with this dimer, nothing worked. The only thing that worked was to take the core of the molecule and place it in my new cell. That was fine, but when I tried to generate the tetramer from the novel NCS, that didn't work either. I end up in a no end of in a refinement hell. So it was clear that there was something else going on here, and of course now we know. And I thought about that. It probably had something to do with the fact that these 4 domains are just linked by one linker, and there's probably some motion that I wasn't taking into consideration. So I tried to use the individual domains as a search model, but that didn't work. They weren't big enough back then to generate enough system. I tried to refine this portion and use the FO minus FC map to manual build in the system. That didn't work either. They weren't clear enough. But what worked was to use the FO minus FC map as a search space in Amore and use the N-terminal domain as a search. So do a phase translation search back then. But that wasn't implemented. That required a lot of thinking from my point of view. And eventually produced a model that had one of the 4 N-terminal domain fixed. Again, I generated the other 3 by NCS, and again, it didn't work. But it didn't work a little bit, it worked a little bit better than that one. And in this case, there was only one domain that was really badly positioned, so I removed that, I did a refinement. And then eventually I managed to position it by hand. And what it turned out is that, of course, of the 3 domains, of the 4 molecules, 3 domains were closed, the 4th one was open. And 3 were completely occupied by ligands, the 4th one wasn't. Now, of course, all I had to do was, if it was today, I would take my data set, I would take my core, I would take my N-terminal domain, feed it to Phaser, and just go have a cup of coffee and come back and have everything done. But back then it took several weeks in order to do that. So in a few years later, I started using MoRep. And one thing that I really liked a lot about MoRep was the fact that I could visualize self-rotation functions. It had a very neat way to do it. And I'm not very good at visualizing things, so having a written-down package helped a lot. And also, it had all this feature in which if you had 3 molecules per asymmetric unit, you could look for the multiple molecules, and they will end up next to each other, and not in 3 different areas of the universe, in which then you had to build, with your symmetry, rebuild the system. Not long after MoRep Phaser came along, and since then I've been using it as like my molecular replacement program by default. That's what, I tend to think that if it doesn't work in Phaser, it will work anywhere else. It might be not true, but that's my modus operandi at the point of view. And it helped me in a lot of different cases, in a lot of difficult cases. One of them was a very important program. I was working at Merck back then, and it was the structure solution of a complex between two proteins. The solution of a structure of a complex between two proteins, p38, which is a kinase, and MK2, which is its substrate. I had crystals that were kind of ugly. They didn't refract very well. The best I could do was 3.1 angstroms. Of the two starting models, one was very well defined, p38. I had several structures. I had been working on p38 for a few years, and I had a collection of structures to choose from. MK2, I only had the PDB. Structure wasn't good. It was about 290-ish angstrom, very flexible, not a very good model. A lot of things were missing. This whole piece at the C-terminal was missing. So the models weren't quite exactly what I was hoping for. And I used phaser. It took, again, a lot of trials. And one thing I learned then was that you have to be very careful in which order you submit your search model into phaser. Because the only way I could get a solution was if I did one p38, one MK2, another p38, another MK2. Everything else failed miserably. And at the end of the day, when I actually got the structure, it was clear that the domains were these are kinases, so the N and C terminal lobe were in a different conformation. A lot of the pieces of MK2 that were missing were actually present, like this activation loop, and most importantly, this p38 loading or docking piece that was also completely missing from the start. So that was a very nice story that phaser allowed me to bring to completion. So a lot of evolution in the model, a lot of evolution in the programs. What about the models? Of course, I think we all know that it's important that we start with the correct model, and we also know that the closer our model is to our target protein, the higher is the likelihood that we get a decent molecular replacement. And we all, in the past couple of days, we saw all these nice references to different ways to evaluate the closeness of the models. For example, the relationship between the RSMD and the similarity, which brings down to 29, 25, 30% similarity, 1.5 Armstrong MSD as line under which the molecular replacement probably won't work. One thing that I think is important, and it was pointed out yesterday, and is that there are very few new fold in the PDB, right? So if we take a look at the fold distribution again in the PDB, this is a slightly different view of what was presented yesterday. In reality, there is no a greater difference. If you take one of this family, I think I did the all alpha, and you dig down, and you see within the same all alpha family, there are different folds within there. But basically, they were all present as a 2000. Everything else, from 2000 on, there is no increase in the type of fold. What is increasing is the number of molecules that belong to the same fold. Which means that if you take any fold, and I don't know, we can take the alpha-alpha superhelix, which is this little thing here, that goes up. Before 2000, there were less than 50 examples of this alpha-alpha superhelix. So there were less than 50 molecules that had that overall fold that is called alpha-alpha superhelix. But of course, they're not all the same, because otherwise they would be the same protein. So they're all a little bit different. And we had a certain sampling on that spacing. Now, 2010, 2012, there are 350 proteins that belong to that fold, which means that we have a much finer sampling of that space, which translates into, if I have an alpha-alpha superhelix, my chances of finding something similar among the 350 is higher than 10 years ago when I only had 50 structures to choose from. Now, sometimes we can't just choose, but what we can do is to take advantage of all this structural knowledge and input it into our molecular replacement programs. And every program does that. You can do a knowledge-based sequence assignment. You can truncate things. You can do alignment. You can use multiple copies. You can use multiple, you can use ensembles that have multiple structures in there. In some cases, and it works. In some cases, doesn't. And just last year, we had a little example of this in our laboratory, and I was talking about, and the program was a kinase. So I think the kinases are the most represented proteins in the database. Only this is a human protein kinase. There are over 3,000 of them, 3,000 structures in the database. And there are not just one kinase, although there is a lot of redundancy. For example, among these 3,000 structures, there are about 190 p38 kinases in there. But there is also a lot of structural differences. So there are serine, threonine kinases, tyrosine kinases, histidine kinases, soluble kinases, membrane-bound kinases. There's a little bit of everything. And so we had a kinase, let's call it kinase X, for which, of course, we didn't have the structure. And we did an initial alignment. Terry Fishman and Corey Strickland did the work. Initial alignment for the PDB, and we found about 30 structures that had between 23 and 40 and 35% of sequence identity. And so we did what you usually do. You look at them, and you pick the one that looks structurally, in theory, based on what we knew, was the more closely related protein to our kinase X. And so we tried everything. We tried phaser, we tried Amora, we tried MoRep, we tried the two loops, put the loops back in, remove the T loop, single domains, domain it together, nothing worked, absolutely nothing worked. So this is clearly one case in which your rational approach is not going to give you anything, or at least it wasn't going to give us anything. But one thing that we have is lots and lots and lots and lots of CPU time, lots of it. And so we decided, okay, if we can't do a rational approach, why don't we try to do a brute force approach? There are so many structural, so many kinases in the PDB. There gotta be one that is somewhere similar to the one we are looking for. So we picked all the kinases that were unique. So of the 190 p38, we only used one. But if there were 2 chains or 3 chains in the asymmetric unit, we used all the chains. And we ended up with about 2,600 search models. We looked in all possible enantiomer space groups, and also at the beginning, just for easy of work, we aligned all the structure on a same reference frame. The automated procedure was Amora to do the molecular replacement. Then there was a packing, rigid body refinement, and then 10 cycle ref MAC. And we started all, and at the end of the day, at the end of a week, I think, I don't even remember, certain amount of time, we looked at the results. And there was one structure that stood out. And this is the R factor at the end of of course it's not great, but this is the R factor at the end of the refinement, and this is the number of trials that were run. And there was one single solution, and it corresponded to a kinase that had less than 30% identity with our target kinase. But at the end of the day, when our target kinase was solved, the RMSD on C-alpha was it was 1.2 angstroms. So really, we are within the numbers. Was it the correct solution? Yes, it was. The density was telling us that was. Throughout the refinement, the kinase was crystallized with an inhibitor, and throughout the refinement, clearly, the density for the inhibitor was coming up more and more, and it was clearly an indication that was the correct solution. So I'm quickly running out of time. I had prepared a couple of slides on the use of different models, different starting models for molecular replacement. Of course, we can use NMR models for molecular replacement. The problems that we have sometimes is that we can't detect the correct solution among everything else, or we get stuck in the refinement. And generally speaking, this has to do with the fact that there are real differences between the solution structure, which is the NMR, and a solid structure, which is our X-ray crystal structure. Differences that are real. But of course, now we know very well how to deal with starting models to minimize these local differences when the overall fold can be the same. We heard a lot about electron density, electron microscopy maps, both as probes to phase X-ray data, low resolution and then phase extension, or the other way around, to use electron electron microscopy maps as envelopes into which we can dock X-ray structures, and so that we can have then quaternary structural information, oligomerization, and things like that. Last but not least, we heard about Archimboldo, so the fact that we can use very, very small pieces now to phase ab initio phasing protein structure and then extend the resolution again. I heard for the first time the single atom phasing, which I thought was very, very interesting, and I'm kind of curious to see where that goes, and we'll see how we move from there. Last but not least, I think we should remember, and we heard about that also in the talk about ampules, the fact that now we have a very good technique to prepare models, theoretical models, or de novo models of proteins that can be used directly into molecular replacement, or we can take these clusters or models and use them as ensembles in our molecular replacement. So I think that we really went a very long time since 20, 25 years ago. One thing that I learned in these 20 years is that a good model is worth 1,000 trials. And a good model is not just a model that is closer to my final result, but it's also a model that is accurate. And so I think that we as crystallographers, but also NMR people and modeling persons, we should be doing everything is in our possibility to make the structure that we deposit the best structure possible. Every refinement program comes with a lot of these structural validation tools, and we should use them a lot, because my structure will be used by somebody else to phase their own data. And I want to make sure that I give to my fellow crystallographers my best model possible. And at the end of the day, I think that whatever works is the one that solves our problem. Everything else is just pure garbage. So I hope we all do our best in order to provide everybody else a good model, a good starting model. And with that, I think I'm going to stop and take any questions. This is a bunch of reference.
Do you have any questions?
If any.
Okay, I have a quick question about your kinase X. You tried single search models in your brute force approach. You didn't try an ensemble approach in phaser.
Yes, they did try an ensemble.
And did that work as well? No. Any more?
Hi, Frank Rundal. You mentioned the GDT, Global Distance Test, as a metric. Can you say a word about that, and how is it more useful for MR, or how do you make use of it?
It's not more useful. I mean, it's a better evaluation of how similar or how different your starting model is to your final model. It's just a different way. Instead of using just the RMS deviation on a single atom, so you basically look at a sphere of atoms. And it just gives you a much better determination of the differences between your model. I think, I don't know if I brought it up there, it's a post-mortem analysis, though. So sometimes you need to know if the model is good before you actually get the solution. So even the RMSD is a post-mortem analysis. So that's good to kind of evaluate it at the end, to say, okay, I used these many models, these were similar or different, and this is a little better way to do it. But it's not, I don't think it helps you in identifying the model that you want to use.
Any more questions? No? I mean, in that case, I'd like to thank Giovanna again. And we move on to Gabor Benkowski from Cambridge, who's going to talk to us about automated molecular replacement with phaser.
Number 4. So I would first like to thank the organizers for inviting me and also for giving me such a late time slot, so I have time to sober up after yesterday's dinner, and also for you who have decided to stay here. most interesting session of the whole meeting. So what I'm going to talk about is a program called Mirage. Well, I do realize that there are several readings of this character sequence, and this is the official one, but you are very welcome to call it any way you like. So what this does, it's an automated framework for molecular replacement. So what are we going to automate? So the ideal way is that we start with some X-ray data, and then without any user intervention, we just have a black box that does all sorts of calculations and comes up with an initial map that you can later interpret. This map does not have to be the best one, but it has to be a correct solution, and we have to know it, that it is a correct solution or not. So I'm not going to talk about any theory of molecular replacement. There were very good talks yesterday about that, but I would like to show you is the logistics, how this can be organized in an efficient way. So what do we want from this program? So first, it has to solve easy cases very quickly, otherwise there is no point for you using an automation framework. The second is, for difficult cases, it should still give a solution, but it doesn't have to be quick, because, well, that's not always possible. We came up with a very simple user interface, or I would say a user interface that is very simple for a very simple case. You only have to specify the sequence and the data. So now I realize that it is not always possible for you to have a sequence for your protein, although in 99.something% of the cases, this is available. If you do not have a sequence, then it's possibly the next talk is for you. But I can't really help with that, unfortunately. If you do not have a data and you are fabricating a structure, then this program is not for you either. However, we allow sufficient customization, so really you can do whatever you like. And I will just show how this works. So now, since this is a de novo solution for structures, then we are optimizing the calculations for a large number of models. And we also want to take off efficient use of the computational resources that are available. This is something I'm going to talk about the next slide. There's one other thing I would like to mention here, just very briefly, that the search algorithm we have is such that it scales with the search complexity. So in other case, if you have a molecular replacement with one peak in the rotation function, one peak in the translation function, and the other one which has 100 peaks in the rotation function and 100 in the translation function, and you have an unlimited number of CPUs, then these two searches, they will take exactly the same time. So, but of course, you never have an unlimited number of CPUs. But anyway, it just scales very nicely. So what do I mean with the computing environment? So first, we allow you to use all the processors that you have in your laptop or desktop machine via the shared memory. However, if you want to go to a larger scale, then you can use a mainframe computer or a cluster machine. And this normally goes through a batch queue system. And there are a number of queues that we are supporting, Condor being the latest addition. So now when I was developing this feature, I realized that, but I would say I was under the impression that this is only for a select few, and myself being one of them. This was not really a bad thing. But later I found out that first, these clusters are not quite as rare as one would have thought. And after I realized that I need a cluster, I found out that in the institute we have one. So in principle, I could use it. The other thing is that in the meantime, technology has moved on. And I would say that up to this point, this is real. What comes next is just for the future. But this queue-based parallelization is so common that people have written software for making virtual clusters in a cloud computing. And so now we are in the UK, we are out of luck, because the government has just turned down a bid from Amazon and Google for providing cloud services. But as far as I know, in the US, there is an agreement with the government. And if you have this software that creates this virtual cloud, or sort of virtual cluster, in principle, you can just run your calculation in the cloud. Anyway, whatever the situation is, one important thing is that a credit card number is going to be an important parameter for molecular replacement searches. And in fact, I will also need one from Randy just for me to allow to implement this. So this is how we actually think of a pipeline. So we are starting somewhere with data, and we also throw in some resources, and we are coming with a linear fashion through to the end. So now this looks very nice as a picture. It's actually too nice to be true, and this is reality. There are so many places that we can skip forward and go back that this single pipe would rather look like some clumsy and patchy plumbing of a British house. So this is not how we are doing this. So now let's see how Mirage is orchestrating the model generation process. So you can specify each of these that will appear on this slide. So there are standard models that are just used as they are, and they go for molecular replacement. However, you can specify just a collection of models that are in some orientation, and Mirage will use the program ensembler to make them into an ensemble. And this can also go into molecular replacement. So now, if you do not have any models, then you can specify a template, and the template normally comes with an alignment to your target sequence. And then there is a program sculptor that will use these two information and put them together into normally a collection of models. There are several protocols in Sculptor that tweaks the model slightly. It cuts the side chains in a slightly different way, and it weights the structure based on the sequence conservation. So you get a number of structures that sometimes they work better, and this is something I'm just going to show later. If you do not even have templates, you can just start with the homology search, and we accept a certain number of formats, and then the program will use the WWPDB to pull the templates and pull the alignments from the homology search. If you don't even have a homology search, we have a way to do a BLAST search for you at the NCBI side. So now, BLAST is not a terribly good method for a very low sequence identity problem, because BLAST is losing efficiency at about maybe 25% sequence identity. So possibly the most common input, I would say, is the homology search. So however, you can specify each at every stage, and the program is using the ones first that are closest to this end. And it is pulling each structure on demand. So in other words, if you have like a 200 hit homology search, the program doesn't spend 2 hours pulling all 200 and then modifying them just to find out that the very first model would have worked 2 hours ago. And yes, indeed, we spent the summer just working out some ways how to generate an ensemble from a collection of models in a rational way, and hopefully this will be available soon. So now this is just the molecular replacement workflow, and I'm not really going to talk much about this, just to mention that this is something you can always do if you have any models and data. However, often you can skip the whole search as it is and just go from the model straight to the solution. So how can we do that, and then what can we do with this? So first, how do we identify solutions? So there was a method that Randy introduced yesterday based on the LLG, and this is an alternative method which is more like, it's more traditional. It's based on the translation function Z-score, but well, I mean, there is a theoretical difference between the two, but technically it's very easy to switch the program from one or the other. And I'm just showing this because it's more traditional, or it's more associated with phasor. So Randy and Early have both showed this table, which was a rule of thumb telling you that which the TFZ score, what chances you have for a solution being correct. And Robert Öffner, who is doing the lunchtime bites, has actually put this into a scientific footing because he did something like 30,000 calculations and then made a histogram out of the Z-scores of the solutions for two important cases. The red is the one when the solution did not work, and the blue being the one when it did work. So you can see that here at about Z-score 6 point something, these two curves, they intersect. So at that Z-score, you have about a 50% chance of your solution being correct. And the higher the Z-score, the higher probability you have for this solution being correct. So now what can we do with this? First, we can calculate the probability we have from a Z-score, and also the probability we want, what Z-score do we need for this. So now if we want like a 99% probability, then we possibly want 7.5, and then we can accept this as a surely correct solution. So this TFZ7 is normally it's a good choice for most of the molecular replacement problems. But if you have a very good models, and this is about 100% identity, then you need to increase this, because you tend to get wrong solutions with these Z-scores. And possibly the expected LLG algorithm does not suffer from this problem, and sooner or later we will switch this program as well. So this is one use, and the other one is that if we got a solution at the very end of the process, we can calculate the probability of being correct from the solution history. The only problem is that the TFZs, as we were calculating it, they do not really refer to the same point. So let's look at this composite structure, which has 3 molecules. And the first molecule we find with a Z-score of 5.5, and then it keeps increasing, which is kind of interesting, because we find a molecule which is the best first. But that's because when we are finding this, we don't know anything about the structure, but when we are finding the last one, we have 2/3 of the structure correct. So to have a proper probability, we just need to recalculate the TFZ, keeping the other part of the structure constant, and just simulate what the TFZ would have been if we found this bit last. And these are the TFZs that we are getting, and then the probabilities are here. And the reason this is not the same as this, because there's a 4th molecule, which I deleted because it didn't fit on the slide. So in the end, we just multiplied the total probability for this composite solution, and then we can say that this is likely to be a correct solution. So say we have found the first correct solution. What can we do with this? The first thing is we can actually stop searching, because we do not want to find another correct solution, because that would just cause more trouble than good. The question is whether we can do this in a feasible way that is also computationally efficient. So the way we normally think about the workload is that first we have different steps, and first we do all the downloads, and then all the editing, all the molecular replacement, and then we come to this identify step. We hit the first solution, and then we stop, and then we go to the refinement. However, by now, we have done most of the calculations, and this identify step is actually a very quick one. So it would be silly not to find these other 3, which we can put together, like Erli has explained yesterday. So it looks like this is not possible, but that's only because we are not attacking this problem in the right way. If we went the other way, so instead of going down, I'm going to the right, then first we do the editing, and then the molecular replacement, and then we hit a solution here, and then we can stop. And the work that we are not doing is not finding these 3 solutions, is something that we haven't done anything so far. We save a lot of time, and then we get the same result. So we stop, but we still want to have an exhaustive result. How can we do that? So say we have found the solution, and we have a lot of alternative models for the same component. If you have alternative models, we can work out a superposition between the 2. So instead of me doing a search, I just superpose the alternative model on the current one, do some refinement, and then calculate an LLG score. In this way, I can very quickly find the best of all the alternative models possible. We can put these 2 things together for working out the search order. So I have this example for this, for an A2, B2 case. And let's say that we have located an A component already. So for the A component, we have a couple of models. We try the first one. It doesn't work. We try the second model, and that gives a solution with a good Z-score. So although we have a third model for this, we are not doing a search, although it would have given an even higher Z-score. What we are doing, we are just superposing all the other models on this, and then calculating the LLG score, and of course taking the one that we had in the first place. So from this, we just select the one that is the highest LLG, and then get a more complete structure. With the other component, that might have given a high Z-score as well, but searching with this more complete partial structure, we would possibly have gotten an even better solution. So there is no point searching at this stage. So now let's see the other possibility that we just try the first component and it doesn't work, and we try the second component and doesn't work either. So in this case, we can just collate all the solutions we have, do some thresholding, do refinements, and then just making up all the solutions we have. And in this case, we possibly have different solutions and also incorrect ones as well. So in this case, we can be maximally I would say efficient and also covering all the search space. So now let's see a real-world case which I got from Pavel Afonin, the author of Phoenix Refine. So this is an A4B4 complex, and we have an A which is 160 residues and a B which is only 110 residues, and they are both very good models available for this. So if we start, well, of course, A is the component that is more likely to give a correct solution. However, if we search with A, we got a lot of peaks and the LLG is not great. If I search with B, then I got less peaks, and although the TFZ is not great either, the LLGs are slightly higher. And in fact, these LLGs are so much higher that the program is not even selecting any peaks from the A. So with this one, the program tries to extend with an A component, and it doesn't work. But when extending with the B component, we get really high Z-scores and a good number of peaks. And very quickly, we end up with 4 B components and a very good Z-score. And from here, it's actually quite difficult to get to an A2B4 composition. And what's the reason for this? And that is in the B factors. So, I mean, these B factors you see are relative to the Wilson B, but it looks like that the Bs, they have very low B factors, but the A components, they have something very high. And that's why, although we are expecting A to be a better model, it's in fact worse. And it's a good idea to try both. So maybe this is just a reference to Giovanna's problem for this P38 kinase complex. So with this algorithm, might have worked out the search order automatically. So what other things can we do? So as we are going along in a solution, the program can analyze the structure that we have, assuming that the structure is correct. So now, one thing that it can very quickly identify is that if a number of monomers, they assemble according to some point group symmetry. So, I mean, right here, it's actually quite easy to see by eye, but that imagine that this is kind of distributed over different asymmetric units or even unit cells. It would be quite difficult for you to see that this is 5-fold symmetry. But the computer would very quickly generate all the possibilities and then come up with a picture like this. So we can identify from a solution that we have a local point group symmetry. So what we can do with this, you can use this information to complete the models, or we can also search with the model as an assembly. However, when we are actually refining the current partial solution, it's possibly not a good idea to keep this rigid because there might be some local differences or deviations from the perfect symmetry, and it's a good idea to leave the different monomers refined as they are. So this is just to show that if we are searching with a larger model, then we get a better signal. So this is a 20-molecule case, which actually has 4 pentamers, and we are searching with a single monomer. So by the time we hit about 10 residues, the program has established that there are pentamers, and it does the next search with the pentamer model. And it's actually finding the pentamer with the highest B factors, but still you can see the TFZ is by far the highest, and also the LLG is by far the highest. And that's because this model is 5 times as large as all the others. So from here, it is really just one more step. If you have established that we have an assembly that is incomplete, it is still obeys the 5-fold symmetry, but there is one molecule missing from there, then all we need to do is just put this molecule where it belongs and just calculate whether it is correct. So if this kind of speculation gives us a z-score and an LLG that is acceptable, then we accept this solution as being right. And again, we can skip the search for this round. So from here, just one more step is that sometimes we know the assembly a priori. So here what I put is actually a kinase. And kinases, they have C-terminal and N-terminal domains. And they have some certain conformation. And normally, it is easier to find the C-terminal domain. So in this case, the program can superpose the kinase and just put the position of the N-terminal where it should be. However, the problem is here that while the other assembly that was kind of determined from the structure solution, so it is very accurate, this is something we just know from kind of a related structure that it could be something like this. So it is not enough just to put it where it is. What we have to do is we have to search around this. And the way Mirage does that is it does a local search. So it localizes the molecular replacement to just a small volume around the position that is coming out from this known assembly. So why does this help? It's actually not faster, surprisingly. But in case you have some large noise peak somewhere else, then these will not disturb your structure solution, and you are more likely to find the correct solution. So yesterday, Deborah was talking about solving the exosomal complex with such a technique that she had a more complete model, which she superposed onto the known parts of the structure she has solved so far, and then so located the positions for the other missing domains. And this is exactly the same thing. It is just automated. So let me just show you this one example, which we managed to solve totally automatically. So this is it's a fairly large protein. It's not a terribly high resolution one, which they solved by SIRAS. So we have about 82 HH-spread hits for this structure. So really, the input is just like that. You specify the sequence and also the HH-spread homology search results. So after 5 minutes, we get a significant hit. So that looks very good. But if you look at the structure, we see that the solution is only partial. But there's an interesting thing to see here is that this homology search hit just covers one domain. And that gave us an idea that we could use the homology search results just to kind of establish domain boundaries. So what we need to do, we just check each homology search result in the CAF database, whether they have an annotation for it. If they have, then we map it onto the sequence based on the sequence alignment that is coming with the homology search. So this is actually implemented in a program phase at a domain analysis. And using this program, this is the domain results that I'm getting for this protein. So you see, it's not actually correct. We have about 40 annotations out of the 80. The C-terminal closest homologs are about 20%. The N-terminal is slightly higher. The correct would be somewhere here. But just based on the fact that this is done only based on a sequence and not by the structure, I would say this is a fairly good result. But anyway, so for our purposes, what we need is just a way to assemble the homology search hits that are for the same component. So the first hit would cover this, and then a second hit would possibly not start from right here, but there might be a little overlap, and this is something we have to allow. And of course, there will be search models that are too small to be useful, and we have to discard that. So using this algorithm, after a little longer, 1 day on 50 CPUs, we get this solution. You can see that the TFZ is not great, but it is slightly higher than 6. But the LLG has gone up, and the solution is actually correct. So, and here I would actually refer back to what I said about the computing power. So this is 1 day on 50 CPUs, and this is scaling very well. So this would have been about 50 days on 1 CPU. So you are not really prepared to wait so much to get a result that is not even guaranteed to be correct. So that's why it's really important sometimes to have the necessary computing power. So, and in the end, we just process an incredible number of models to get this solution. I'm not saying that they could have solved this structure with molecular replacement, because I mean, if I started auto-building from this result and not using MR Rosetta, auto-build just came up with this model, which is about 50% complete. But you can see that the part here that was not present in a model is kind of nicely put back, and also the crab that was here that is nicely removed. So that is hope. Just very quickly, a benchmark about phasor, so how we can really improve a result slightly bit. And what this graph shows is the success of phasor, or well, molecular replacement against the sequence identity. So the blue curve is the one when we do actually find the correct solution. It is just not the top one, and there are many other ones. And the red one, when there is only one solution, and that's with a high Z-score. So, I mean, there is a little bit of a difference between the two curves, and we are working hard to kind of push this red one onto the blue one. So, just made a very quick benchmark, which is again based on the calculations from Rob, and we defined the input as cases when phasor has found the solution, but it was not the top solution, which is a subset of the cases that I showed before. So now we are running these searches again, but not just using the first model, but all the models that Sculptor is generating. So in this case, this is showing the distribution of the benchmark set against the sequence identity. So you can see that we have a relatively large number of models between 20 and 30% identity. So this is just the height of the columns. So initially, we start with all these columns being green, which means unsolved. However, if we switch on all these Sculptor template models, the blue ones indicate solutions when there is just one solution and it is a high Z-score, so it's very easy to identify. And the red one indicates cases when the top solution is the correct one. So these are the results we get. You can see that we actually managed to get rid of most of the green one, and well, not all of them, unfortunately. But this is still a step in a good direction. So this method seems to work very well to about below 30% sequence identity. And that's because if the sequence identity is higher, it's more likely that the structure has a systematic error and not this kind of random error that we can easily compensate for using different b-factor weights. And all left is just to acknowledge the whole phasor team are all here, including Andrea, who has just joined the group, and the fruitful collaboration with Phoenix, and then all the advice we get from the industrial consortium, from the CIMR people, and also from CCP4. And this is just the acknowledgement of the Phoenix consortium as well. Thank you very much.
Questions for Gabor?
You mentioned the CPU time, but what about GPU?
Well, well, I was actually taking just the easy way of parallelizing the molecular replacement. So for GPUs, that would require a lot more in-depth restructuring of the algorithms. And it would possibly also not be so transparent, so that you would not be able to run on clusters as well. But yeah, this is certainly a way to go ahead. And I mean, right now it is debated whether GPUs would help science or not. So we are taking some sort of a defensive stance and not moving until this has become clear.
Okay.
I cover the conformational thing that Giovanna mentioned, which was an explorer where you can generate different models with different angles. Is that no longer relevant with more sensitive detection techniques with smaller models now, or is that something that's worth putting into your decisions?
Yeah, definitely. So, well, yeah, that is certainly a good one to do. I mean, right now, I think phasor used to have such a functionality, but I think it's no longer present. So, but we could definitely reinstate it. So, good. So then it's easy.
Any more? Okay, I'd like to thank Gabor again. And our next speaker is Gertrude Spitz from Harvard, who's going to talk to us about tools for disparate crystallographers, should such beings exist.
Which computer is it? Is it 2? Thank you very much for the invitation, and I'll take it, I guess, one step further. We won't ask for sequences. We'll just take MTZ data, and we'll have a couple more computers to run some of our computations. Thank you very much for the invitation to the CCP4 group, and on behalf of this SBGrid consortium, I also wanted to say that we are very thankful for your support of the application to the community and for the workshops you guys been doing in the United States. So this is the overview of my presentation. Let me just get the pointer. Better pointer. So wide search molecular replacement in general, it's something that might be useful to a limited set of people. It's something that you will typically try in cases where you try other approaches. Typically, people who, for example, crystallize something when they don't have the sequence might be able to use it, but also in the cases when you don't have models with high enough sequence similarity in the database, you can try to do it. Potentially when you crystallize a contaminant, this could be very useful. And in addition to users, I think is also a valuable tool for developers to kind of look how, on a global level, metrics that they use in the applications are responding to different scenarios. So what I'll do in the talk, I'll briefly provide you the definition of the wide search molecular replacement. I'll go over 3 examples with the multi-domain protein complex, single domain complex, as well as DNA, RNA structures. Then I'll tell you what we've learned from those examples in terms of scoring metrics, as well as in terms of selection of potentially the best model that can be taken to the subsequent structure refinement or modeling. And we'll extend it to the examples showing how this can be useful for evaluating the metrics in different applications. And in particular, we've done the recent evaluation with a subset of the domains from the PDB redo project. And the question is if those redone, re-refined structures are any better for the molecular replacement. So we've kind of done this evaluation on the global level. And finally, I'll show you how to submit, if you wanted to try this approach, how to submit the computations to through the portal at Harvard Medical School. So the principle, it's fairly simple. As I mentioned, you just need a data set. You don't really need to provide a sequence file. And the approach of wide search molecular replacement relies on computing the molecular replacement solution with many, many models. And this is not dozens. This is really thousands of models that you're putting in the pipeline. You can use molecular application of your choice, pretty much. You can do it on your own computer with many models. And when you obtain the results, we don't look at them individually. We create a global graph, and we try to basically compare the positive to negative, what the decoy set is doing, basically defining the boundaries of the solutions that might be correct. So the particular implementation, which is currently in place through the portal at Harvard Medical School, it's using the SCAP database, and this is 100,000 domains. So basically the PDB chopped down to individual domains. We use application phaser. All those computations, so we submit 100,000 molecular replacement jobs to the what's called Open Science Grid, and this is the large computational grid in US. We use it opportunistically, and basically on our side, it doesn't require any maintenance. What we do, we submit those gliding jobs, which are going to the gliding factory in University of California in San Diego, and then those jobs are kind of pilot jobs are dispatched to the different computing resources, and when they are on the top of the queue, they report back to us. So dynamically, very quickly, and for free, we can build up a cluster of 10,000 CPUs and basically have it available for our computations. So we submit those phaser jobs, and typically once the job is submitted, we get results back within 12 hours or so. So this is 100,000 phaser runs. And then we construct this graph, which is basically LLG versus TFZ. and we try to identify if from this graph we really get anything which is worthwhile for structure determination. So let me give you a couple examples, and the first one is with the MHC TCR complex, and this is something which is in particular very interesting because of the composition, domain composition of this complex. We have the peptide binding domain, which is unique to this in the complex. It's about 22% of the molecular weight of the entire complex, and we have 6 IG domains, and individually they contribute each about 12.5% by molecular weight to the entire complex. When you look in the protein data bank, or when we've done this exercise, there is about 600 peptide binding domain candidates in the database that could be potentially used to solve the structure, and we have close to 5,000 IG domains in the database with ranging degree of sequence identity that could be used. So we run this wide search molecular replacement, and this is the solution. This is what we get. And ignore colors for a second. We really have this one big set here of what we potentially suspected are the solutions. And if you notice here, the TFZs range from 12 to close to pretty much 4, 3. And all of them have elevated LLG values. And then we have this second subset, which we suspected are not the correct solution. So when we analyze this top subset, it happens that all of those domains here marked in red correspond to the peptide binding domain. So this is kind of a really nice validation that in the case when you have 22% of the structure, you can effectively get many, many solutions. And can it go even lower? So when you look at some of those gray peaks here, those actually start to correspond to the individual IG domains. So you can even go as low as potentially 12% in this case if you select the right domain to get the significant solution which will allow you to, which can be identified. So about 50% of those remaining red dots are the peptide binding domains which have lower scores. So the question right now is, so we have two distinctive subsets of the peptide binding domains, but do the higher scores really represent the correctly placed domains? And we've done the analysis using some of the CCP4 tools, and indeed, and this was really amazing, that all of the ones which have high scores are correctly placed. So effectively, we don't have any negatives. None of the red ones were correctly placed. So this is fairly encouraging, and it really speaks to the power of the both TFZ and LLG scoring. So just kind of a side interest I wanted to show. So in principle, if you, for example, place 3 of the IG domains, and we could have done it with the search and calculate density modified maps just based on those 3 components, so it's only was 36% placed, we start to see proper densities for the remaining parts of the structure. So this is electron density map calculated with Pirate for the peptide binding domain. So using this approach of slowly placing parts of the structure, you could build it up. So that's another example of flavor protein, and that's a single domain search right now. So this particular structure initially has been solved by heavy metals, by heavy atoms. When we do this search with wide search molecular replacement, we get about subset of 7 to 8 peaks, which are really strong. It could be easily identified by TFZ. Then we have a smaller subset of things which start to have TFZs closer to 6. They have very strong LLG values, so if you try to identify them by the combination of 2 metrics, you can do it. And then we have a couple of really weak solutions, which you couldn't identify by TFZ, but what Randy was talking about yesterday, if you really look at the values of LLG above 40 or close to 40, you could potentially identify them. So in this particular case, all of those domains, so the red ones are, again, the flavoprotein domains which weren't placed correctly, and they buried. So out of the subset of about 200 domains, we really get a handful of them which Phaser can correctly place. In terms of the sequence identity for this subset, it's fairly low. It's between 5 and 25%. So we're getting fairly far from the ideal model. And we've tried to initially validate them by just refining them, and effectively none of them could refine using standard RefMac, Phaser, Phoenix, etc. tools. If you run Phoenix auto build, however, for all of them, even the ones with the lowest sequence identity, we very quickly arrive at the refined at R3, which converged close to the values that were reported in the deposited PDB. So that's another set, and that's just an example of the shifts which you see in the structures. This is the model, the top model actually, which we identify in the search, and this is the target structure with the electron density map. And if you calculate density modify map, the map will converge on the target structure. So the latest addition here was RNA and DNA models, and we downloaded the X-ray Atlas database from Rutgers, and we run it against this 1 angstrom structure of the decamer, DNA decamer, and that's the graph that we're getting. So in effectively, it's very similar to the protein landscape. However, this is the original PDB file from the target data set, and we have a number of very strong solutions. All of them will be correct, but notice here the scale for the LLG and TFZ. The values are not quite the same to what you use when you're working on the proteins. The LG values are way above 500 for the correct solutions, and TFZ values are also stronger. So we haven't completed this analysis yet, but it just indicates immediately that maybe the scaling is a little bit different in the DNA, RNA universe than it is in the protein cases. So that kind of goes up, doesn't fully agree with the recommendations on the Phaser website in terms of what is the possible and probable solution. And that's just looking at some of those cases, just confirming that they correctly placed. So what have we learned in terms of the scoring metrics and placement of the domains from those examples? And I think the major thing is that when we look at the TFZ scoring, this traditional recommendation to look at things above 7 certainly is correct. Everything above 7, majority of the things above 7 in the cases that we've done are correct. However, we see cases which have TFZs as low as 3.5 or 4, which are also correct. So those could be equally well used to determine your structure. Obviously, they mask by a lot of solutions. In this case, a lot of incorrect solutions which have TFZs as high as 6, or in some cases even approaching 7 or 8. So if you're just looking at the absolute score, you could be potentially fooled taking one of those solutions, working with them, and they wouldn't really help you with the structure determination. So what we found really useful is really combining LLG score with the TFZ score. And as you can see here, they correlate really nicely. By looking at the LG scores, you can certainly fish out those solutions with TFZs as low as 3.5 or 4 and suspect that they might be correct. And when you're looking at this analysis, you can kind of try to cluster the top peaks and check if they correspond to the same fold, and this would be another confirmation. So do we need to run many cases? Do we need to run cases that fail? And potentially, I mean, you've heard in the previous talk there are ways to limit, et cetera. But in this case, what we gain from running those cases which don't converge or are false, we really kind of, without doing complicated calculations that Randy mentioned yesterday, we've kind of getting the boundary or the level for the particular structure of what is correct, what is incorrect LG. So from this surge, you can see that here, maybe around 37, 38, it's a cutoff for what could be potentially interesting for the structure determination. So again, looking at this flavoprotein structure, kind of similar landscape. We still have things which you could pick up by TFZ, but combining both of them, and in particular looking at this boundary of about 37, 38, in this case, maybe even lower, down to 30, would allow you to identify some of the weak solutions from this search. So that kind of underscores the value of the negative subset. So further, we can also look at other metrics, and in this particular case, we're looking at the rotation function score. So RFZ score from Phaser works equally well for identifying correct solutions in combination here with LG. And you could potentially further combine it with other things. So this is now TFZ, RFZ, and LG all used here, color-coded. And from this, you can identify the top solutions. So you can kind of extend it by looking at other things in a global way, try to figure out which metrics are the best or the most sensitive to the correct placement, correct solutions. So the second question is, for a user, by this approach, by LG, if something has the highest LG score, highest TFZ score, is it helpful to take this particular model and then use it in the refinement? Do we get better models basically selected immediately after you run the molecular replacement computations? And this is again looking at the MHC TCR complex at this first search that we've done. And what you see on this graph, I'm plotting RMSD of a individual SCOP domain against the target molecule versus LG. And what we can see from here that we have a fairly good correlation. Effectively, the domain with the lowest RMSD has the highest LG score. So that then translates here to the molecule which is closest to your target. So obviously, if you have a difficult case when the auto building or refinement doesn't converge, it would be worthwhile to have something which is closer. If you just look purely at the sequence identity, for example, we have the case here when we have many, many domains which have 100% sequence identity, the LG could be fairly variable for this case. So you could do it based on sequence to a certain degree, but certainly LG itself, it's a powerful enough metric to allow you to identify the closest model to your target structure. And that's looking at the flavor protein case. And interestingly, in this case, the things which had the lowest RMSD, so I mentioned this one has a lower sequence identity, and the RMSD range is basically between 2 and 3.5. And you can see from this plot, again, LG versus TFZ, that things which had the lowest RMSD actually failed in the search, and that's maybe due to the overlaps or some other problems, but things which were just immediately behind it at about 2.2 do really get the highest scores. And here you look at this plot, you basically have the range of RMSDs between 3.6 and 2.2, and the green ones are the ones that gave you the correct solution. They kind of cluster towards top of this, the lowest RMSDs, but there are several ones which fail to provide correct solution. But in general, they are in this range. So we do see not as great correlation as in the previous example, but it's still fairly strong correlation in this one between the RMSD and LG scoring. And this is now in comparison based on the sequence identity. That's not as great. It's much more random. We do get the range between 12% in this case and 20% when we do get correct solutions. And actually, just go back for a second to the previous slide. This I think valuable lesson here is that the range of convergence of molecular replacement in this case, we can hit things with RMSD as low as 2.7. So anything between 2.7 and 2.2, it's a fairly fair game for this particular case. Anything below 2.8, we didn't get a single solution in the molecular replacement search. Okay, so what about examples? What can we learn from kind of development perspective? So I think we've done this particular case for Randy a couple of years ago when we went to the new version of Phaser, and I think they tried to improve TFZ value, and the scores got higher, but when we do our test, they kind of uniformly get higher for both the correct and incorrect solutions. So there was some correction in the computations, but globally, when we do this analysis, we don't really see much help from the new way that things were computed in terms of identifying the correct solutions, because you have this global shift. This is for MolRep, and we've used MolRep extensively. I've solved a number of structures with MolRep, and it has been very productive for us. In comparison to Phaser, so this is again for the peptide binding domain. And remember, in the case of Phaser, we have 250 correct solutions. With MolRep, we actually get 20 more. So we get 270 correct solutions using peptide binding domains. The problem with MolRep, with the particular version we tested 2 years ago, is that you don't have this nice discrimination you had in Phaser between the correct and incorrect solutions. So when you use the MolRep score or contrast function, there is a significant overlap here between green and red. So you couldn't use this metric to really identify what's correct or incorrect. However, you could pretty much assume that you will get the correct solution by just running MolRep computation. So that's another case we've done is with I mentioned with the PDB redo data set. And you guys are familiar with the project. The question, I think this is the title of the paper, constructive validation, more they're just looking for errors, right? And I think in the paper they actually show a couple of cases of refinement. However, are the models better for the molecular replacement? And we've taken a look at this flavoprotein data set. So this is the subset I showed you before with about 200 domains, which potentially could give you the solution. And after refinement with PDB redo, so out of the 200 data sets, about 136 domains were done by X-rays versus NMR, and they had diffraction data sets. We didn't need to have diffraction data sets for this data set, but PDB redo, in order to improve the refinement, obviously you have to have diffraction data. And those 136 domains come from 86 unique PDB files. So globally, PDB redo improved by about 2%. So those are the improvements in the R3 for the individual structures in the subset. You can see the drops between the PDB and PDB redo, 2 to 3%. In case of RMSD, the changes in the RMSD between the PDB redo and PDB subset are between 0.3 and 1 angstrom. So we run this first test, and this is with PDB data bank subset of domains. We get about 23 hits. We get similar number of hits from PDB redo subset. We do get a slightly better separation between correct and incorrect, so maybe minor, fairly minor difference, but potentially some small improvement. In terms of LLG changes, really one structure got a significantly better score. A couple of them actually got weaker scores. So again, mostly cosmetics. Some interesting things when you look at the top peaks. Actually, the top peaks in the PDB subset came from the same structure. And so you can see probably what happened in the PDB redo refinement, the NCS restraints were released. So right now we have a slightly bigger spread between those four top domains. They still score with similar values of LG and TFZ, but they don't overlap as much. And then what we do, we run it as a function of resolution. So we start basically the search all the way from 3.5 to 10 angstroms. And 3.5, 4.5, it's about the same. And this is using PDB data set. At 5 angstroms, we suddenly got some artifact. The TFZ is scoring fine, but LG, so this is the top solution that finally lost the LG value at some artifact of the particular version that we used. You go to 6 angstroms, you pretty much lose the boundary. So those will be the correct solutions, those will be the incorrect. So you now move to 6 angstroms, 7 angstroms, you don't have clear boundaries. And even if you go all the way to 10 angstroms, you don't have those clear boundaries. Suddenly you have a really nice peak in a TFZ value with really low LG. And if you look at this particular domain, this will be the same as you had before on the top of the search. So it's really some kind of peculiar resolution dependence. So now if you look at the same with the PDB redo data set, it's pretty much similar at 2.5, 3.55 angstroms. At 6 and 7, we get still much better separation than we get in the PDB set. So there is some difference between two of them. It's not significant, but in this case, PDB redo seems to give slightly better results, especially at the lower resolution. And again, if you go to this 10 angstroms, we had the same effect as we had with the previous data set, the high TFZ. And Randy is shaking his head. It's no idea. It's just, I think we talked about it briefly yesterday, that it seems that the really low resolution LGs is not fully optimized yet. But this had been with the LG score. So now if you just want to submit the computations, you just go to the SBGrid portal at Harvard Medical School. And for those of you who are not familiar with SBGrid, this is a consortium of about 220 academic labs which collaborate to maintain the software infrastructure. The portal is operated out of this website that has been supported by the National Science Foundation, the development of the portal, and is available freely to all structural biologists. So you don't have to be the member of the consortium to come and use it. You click here, then you get the second screen. You launch the WSMR computations. You have to register, create an account to be able to come back and look at your data. Once you created the account, you create a job. And this is pretty much all you have to complete in this window. You give the job some name. You have to justify why you want to use WSMR. We won't run all the submitted jobs by default. You have to tell us that you suffered and that you tried other approaches and why this particular computational approach might benefit your case. We might use it to set up priority or potentially turn you down, but in general, if you have justification, we would eventually run your jobs. You just upload the MTZ file. You tell us the columns F and FP. The rest of this is default. As you notice, no need to upload the sequence. You can put the sequence. It will help us with the analysis post-mortem if you see interesting results to potentially check if there was an obvious hit, but it's not required. And you can select one of the databases. This is the SCAP by default, 100,000 domains, but we also have FAB data set and smaller sets of the SCAP database. And if you want to check for enantiomorphs, you can click here. You can also put additional phasor parameters in this optional box if you wanted to put them into the equation. And then you wait until the job distribute on the grid and come back, and you get an email when the results are ready. And then you will get graphs. Pretty much you get two graphs out of it. The first graph will show you the spectrum you got from your results, and then the second graph you can launch. You can click on individual data points and look at the individual domains. And after this, the things aren't automated. You pretty much go and download the SCAP domain to your own computer. You try to rerun the phasor, make sure you get the same computations, and you then go off with the PDV, with the Phoenix AutoBuild or Rosetta or other approaches that you might want to do to validate the correct solution. So just at the end, I wanted to show you a couple. I've just looked yesterday. We haven't really actively analyzed the cases that have been run for the last several months, but just yesterday I went and looked at some of them. So as you can see, for example, here, looks like somebody got a fairly strong solution. The TFZs are way above 7 for the majority of the data points, and the LLG values are pretty much, you would say, more than 50. Those might be potentially correct. There is another potentially good solution, and this one is a little bit more classic. So the LLG is below 40 for the full data set. This probably failed. This maybe has some correct solutions here. You would have to really look and try to cluster, make sure they come from the same family of domains. This is very clean. This one potentially has a correct solution. This one, you get something really high with LLGs of 200, TFZ 20. This one, again, the correct solution. This one is obvious. This one, potentially. I looked actually at some of those domains belong to the same cluster, so that is probably a correct solution as well. This is a very nice spread, and you probably want to use this one for molecular replacement. This one, it's not obvious. Maybe it didn't work. Okay, so future opportunities. I think for you as a community, and I think this is a major lesson, based on what we see, you can really get correct solutions with low sequence identities, low structure coverage, low scores. With all the new tools which are being discussed in this workshop, I think there is a tremendous opportunity to take those weak solutions and then convert them to the correct structures. And using any of the new tools that have been discussed, Phoenix, Rosetta, morphing, auto-building, ShellXE, etc., you can do much better than we've been traditionally doing in past. For developers, I think it would be worthwhile to think about the standard test sets which are used by the community to really track the progression of the methods. To check, for example, with this domain that I show, we can only get right now 12 out of 200. With time, are we doing better? And by using different tools, can we really push the boundaries? Because right now, I think especially from the user perspective, I think there's a bit of confusion, and with so many methods, they clearly work for individual particular cases, but the sense of what is really the robustness of them overall, I think it's something that is currently missing. Also, from the developer's perspective, we've developed this opportunistic access to the OSG resources. And Pavel mentioned in the previous talk, you can pay for the cloud services. This is something which we actually don't have to pay, so you don't have to have a credit card to use. But it can provide you with really access to thousands of computers. If you want to try new approaches, not necessarily for production, but just for testing new algorithms, new cases, you are welcome to talk to us to use it. And for us on the portal side, we certainly need to put new databases in place, update the databases. We already have FAB in place, maybe extend it to using automatic model rebuilding tools, and potentially test new parameters. So if you have any questions, just email wsmr@sbgrid.org or just go to the website to submit the jobs. Those are the references. We had a paper at PNAS back in 2010, and we're trying to prepare the paper for this symposium. Those are other papers. And finally, I wanted to acknowledge members of my group. In particular, Ian Stokes-Rees developed really most of the portal technology and initial WSMR approach. Daniel O'Donovan took over after him. And he also developed low-resolution DEN refinement in collaboration with Axel Brunger, when we can go and perform low-resolution searches using Portal. So that's also available. And now we have Mick Timoney and Jason Key, who are both in this picture right now. It's Jason, who just joined from Kevin Gardner Group and from Dallas, and they'll kind of continue the project. Support has been provided by the National Science Foundation, as well as Open Science Grid. And this is my happy group ready for the holiday lunch a couple of weeks ago. And part of the group is doing the wet lab. We're working on the microRNA biogenesis. And this has been really nice collaboration with the lab and discussions when we can take the wet lab projects and talk to the computing guys. It's difficult to make them really connect well, but we've been trying, and we make some progress over the last year. So thank you for your attention, and I'll take any questions that you might have.
Questions, Piotr?
Frank
Maybe you can go with the microphone.
The search models, have you curated them in any way, or are they just the PDB stripped of waters? So have you cut them down to cores, or?
So we've curated them to a certain degree, but effectively we're using the SCOP database, which are the cut down PDB files. Some of them had problems in terms of the B values, etc. So we try to correct for the obvious errors, but we don't do any kind of intelligent truncations and things like this.
Tom Terwilliger. First, a comment on the fact that PDB redo those structures didn't improve your success rate. In retrospect, at least, makes a great deal of sense. It would be amazing, in fact, that if that had worked, because the starting structures are pretty accurate, right? These are all good structures. They're all in the PDB, basically, right? And so PDB redo has improved those very slightly towards their true structures. But it's not improving them towards
the
target, which is very far away. So the improvement in the direction of the target is going to be exceptionally small. So I wouldn't expect any other result. It would be amazing if it turned out otherwise. But in taking that further, another thought, so you have this amazing amount of computer power that's available to you. What about the idea of taking, for example, many Rosetta models for a structure and just running them all? And that those Rosetta models will show a great deal of diversity, and sort of by chance you might hit some good ones in a way that's orthogonal to the way that you're already searching.
So that's one option. And another one is take, for example, MD trajectory, run the MD simulation and capture many frames and check if any of them by chance matches your structure. So I think that's certainly something we're talking about. Ronan?
I've got a question.
Yeah. Oh, sorry.
I have a question on the logistics of accessing this facility. If on the last slide you mentioned that there are smaller subsets like FABs or maybe the MHCs and things like that. Would that make my access to it higher priority, for example, because it takes shorter time instead of having to do the 100,000?
Yeah, potentially, because that's a job we can kind of squeeze in between the other jobs, and they'll run 30 minutes. One thing I should mention right now, it's not available to pharma because of the Open Science Grid is an academic collaboration. So there is a couple local resources. We can potentially set it up for pharma, but it would be much longer. And the threshold to run it will be higher.
Graham Winter, Diamond. I have two questions. Roughly how much CPU time does it actually take to do one of these jobs? What order of magnitude?
Right. So we're talking about really depending on the cutoffs, and it could be between minutes and I think at most 20-25 minutes per job if things don't converge.
But that's 25 minutes times 10,000 CPUs.
For the really cases that don't converge, I think on the average we converge in about 1-2 minutes per SCOP domain.
Okay. Because what I was thinking is, I mean, would this be something that a facility could actually install locally?
If you have the large computing cluster...
We've got a couple of thousand cores.
Yeah. So basically you have what we have with Open Science Grid without all the complexities that we have to face by submitting the jobs to the grid. So you can certainly, I mean, effectively you can just do your own implementation of the wide search molecular replacement. It's just phaser wrapped to run with many models, right? So that you could certainly do it if you have a couple thousand CPUs.
Okay. Thank you.
Robert from Cambridge.
Okay.
I guess that the LNG values and the TFZ values you presented, they weren't the refined values after the end of the calculations, because there were some negative LNG values ever so often among the four solutions, correct?
Yeah, so we take just the final value from phaser, and this is version 2.3.
Right, okay. So you probably talked with Rand that the current version of phaser.
Right,
yeah.
we've shown and it can normalizes, etc. So
yeah, because then it should be a lot clearer which are the correct and
right.
incorrect solutions.
Yeah, so that would be maybe one of the things to do for this post-conference discussion to figure out we're doing better with the new version of phaser.
Right.
So, Pietro, in the context of the questions as to fold coverage and how often do we have a protein that has a known fold but we can't tell from sequence, do you have any sense of how many people using it on a sequence that they couldn't really think they could use molecular replacement on because it didn't really resemble anything in the database, ended up hitting a solution and it turned out to be
I wanted to go over those examples, and we'll probably find out after we do analysis. We have a couple of cases when people submitted plant proteins, and they said specifically, we purify from the plant, we don't have the sequence, and this will be really useful for us. And I look at a couple of them, and we have nice hits in those cases. But I mean, that's really the holy grail of this setup, if we can hit the things which don't have the identical domains in the database, but still come up with smaller fragments. Could be potentially then extended by shell-X, etc. So that would be really neat. But we'll look at those cases. I'll maybe in the paper we'll let you know what was the success rate or which cases worked.
Okay. Any more? Okay, I'd like to thank Joshua again.
Okay.
Our final speaker is Andrew Cruz from Stanford University, who's going to talk to us about the application of molecular replacement in G protein coupled receptors.
Okay, can everyone hear me? So to start off, I'd like to thank the organizers for the opportunity to be here and speak. It's really a privilege to see everything that's going on within the computational crystallography field. This obviously affects us a lot as users of these programs and everything, to see what's going on and what's going to be available going forward. So today I'm going to talk about applications of molecular replacement to G-protein coupled receptors. And before I begin, I think it would be good to give a brief introduction to just what exactly a G-protein coupled receptor is and what it does and why we should care about them. So G-protein coupled receptors are transmembrane proteins. They're located within the plasma membrane of virtually every cell in the body. They play a very important role in regulating pretty much every physiological process. This includes things like blood pressure, heart rate, neurotransmission, digestion, as well as the senses of sight and smell. So they're very important proteins, and they work by communicating a signal from outside the cell across the membrane into the cytosol. They do this through conformational change, resulting in interaction with G proteins or with other effector proteins, such as arrestins. In general, GPCR signaling is very complex, and it's all regulated at the level of conformation. And so in the Kibelka lab, we're very interested in understanding this particular aspect of this process, which is the way in which conformational changes in the receptor are induced by ligands and how they lead to interaction and activation of intracellular effector proteins such as G proteins, arrestins, and others. So I'm going to go through today several of the recent advances in GPCR crystallography and how they relate to molecular replacements and the implications of these advances. And then I'm going to go through a series of 3 specific cases of structures we've solved by molecular replacement. The first case is the mu opioid receptor, which represents a fairly typical case. It was solved relatively straightforwardly. Then I'll go through the M3 muscarinic acetylcholine receptor, which was a much more challenging problem, particularly at the level of data collection, and it highlights some of the difficulties that we often face in solving GPCR structures. And finally, I'll go through the beta-adrenergic receptor GS protein complex, which is an example of a multi-protein signaling complex, which is solved by molecular replacement using individual components. So in the last several years, a huge number of GPCR structures have been solved and reported. To date, there are 18 unique GPCRs for which structures have been published, and I've shown a subset of those here. You can see by structural alignment that they actually show quite a high degree of structural conservation, particularly within the central part of the transmembrane bundle. And this is actually kind of surprising, I think, because if you look at the sequence identity, typically pairwise sequence identity for these is in the range of 30%. In some cases, it's even much lower. It can be as low as 20% for some of the receptors shown here in this diagram. And in spite of that, they really are quite similar at the level of tertiary fold. It's also worth noting that all of these structures, with one exception, have been solved by molecular replacement. The single exception is bovine rhodopsin, which was the first GPCR structure to be solved. And because of its high its high structural similarity to other subsequent GPCRs, it's been possible to use that as a molecular replacement model and then subsequently use other structures derived from it. So one of the major advances in recent years in GPCR crystallography has been the use of lipidic cubic phase methods or mesophase methods. This is something that's been particularly advocated by Martin Caffrey, who's developed a variety of techniques that make this much more tractable. This is a technique which involves first reconstituting the membrane protein in a lipid bilayer and doing the crystallization with the protein embedded in the lipid the entire time. This is usually performed in a glass sandwich plate setup, which involves suspending the sample between two layers of glass separated by about 1/10 of a millimeter. And because of that, it's very difficult to do things like heavy atom soaks because we can't open up the plate and then seal it again. Once the plate is open, we have to break the glass, and then the only choice is to remove the crystal. There are some other methodologies that are more tractable for experimental phasing in LCP, but in general it's challenging. So this is one of the main reasons why we're really dependent on molecular replacement, at least as we do GPCR crystallography in our lab. One of the other major implications of this is that the crystals always grow containing a bilayer, because the protein's in a bilayer and the protein is in the crystals. The crystal itself necessarily contains a bilayer within it, and that organization is useful as a really quick check of whether or not a molecular replacement solution makes sense. So if we get an ambiguous solution, we're not quite sure if it's right or not, we can just take a quick glance at it. If it's from LCP crystals, we will, at least so far without exception, see this kind of a packing. So this is showing layers of receptors stacked on top of each other with intervening layers of aqueous phase. And this stands in contrast to the so-called type 2 membrane protein crystals, which are commonly seen for membrane proteins crystallized in detergent. And this has sort of a less organized arrangement, not in this sort of stacked layers, but rather creating spaces to accommodate a detergent micelle. So this packing that we expect to see, this type 1 packing, is really useful as a very quick way to test whether or not molecular replacement solution seems at least sensible and reasonable. One of the other major advances in recent years has been the use of fusion proteins and crystallization chaperones. So I've diagrammed here the T4 lysozyme strategy. This is a method that was developed by Dan Rosenbaum, and he was working on the beta-2 receptor, which has a very large, very flexible intracellular loop here. And what he did is he replaced that loop, deleted it entirely, and replaced it with T4 lysozyme, which is a very well-ordered protein. It's an extremely crystallizable protein, and this provides a large amount of hydrophilic surface area, and it also can provide phasing information to facilitate molecular replacement searches. This technique ended up giving this structure here, which was one of the first structures of a human GPCR. So in some cases, we also use crystallization chaperones, things like antibody fragments shown here. And these can be quite useful for molecular replacement because they're often quite large relative to the receptor. And as such, they have a fair amount of phase information associated with them. So we can do a molecular replacement on a known soluble component like a Fab, solve the structure with that, and then use that to build in the rest of the receptor. Although in general, we don't actually need to do this just because the receptor is so well conserved. So now I'll go through a series of 3 specific examples. To start, the mu opioid receptor. This is a receptor that's playing a critical role in regulating perception of pain. It's important as the target of morphine and a variety of other drugs. it's also important as the target of a lot of drugs of abuse, including heroin. So we were interested in solving the structure, and Ashish Manglik in our lab set about trying to crystallize this protein. He was actually able to obtain crystals quite quickly, and he obtained 2 sets of crystals, one set that looked very nice and diffracted very poorly, and another set that looked terrible and diffracted very well, at least relatively speaking. And so from these strongly diffracting crystals, by our standards, we were able to compile a complete data set to 2.8 angstroms. And you can see that this comes from 25 crystals. This is very typical of the way that we do data collection in our lab. Because the crystals are grown in LCP and they're very small, they're very fragile, they're very radiation sensitive, we typically require a large number of them to create a complete data set. We can usually only get something on the order of 10 degrees of data from a given crystal before radiation damage becomes so severe that we can't get useful information anymore. So based on a Matthews calculation, we anticipated 1 to 2 molecules per asymmetric unit. I should mention this is also an example of a T4 lysozyme fusion protein, so it contains a receptor and a T4 lysozyme molecule within the asymmetric unit. So to do the molecular replacement search, first we chose to use the most homologous receptor that had been solved previously based on sequence identity, and that is the CXCR4 chemokine receptor, which was 27% identical in the region that we were looking for. We did the molecular replacement search in phasor, also searching for 1 T4 lysozyme molecule, which is, of course, 100% identical to the search model. And then finally, when we get a solution, we can ask, does it make any sense, just based on TFZ scores and so on, but also based on what the packing looks like. So as I said, this was actually a fairly straightforward case. This is usually what we see when we solve a new structure in our lab. We got a solution almost immediately. We see receptors arranged in pairs that are stacked in layers. This layer here, shown in cyan, these are receptor molecules. This would be filled with lipid. And then the green layer of T4 lysozyme molecules is an aqueous layer, and it contains water and solvent. The TFZ scores for both components were quite high, just a little bit over 7. And as I said, the molecular replacement solution shows these stacked layers, which is really reassuring as an indication that the solution at least is sensible. We can also further validate this just by looking at the position of the T4 lysozyme, which, as I said, is inserted into the third loop of the protein. So we know that the N and C termini have to be in close proximity to the junctions with the receptor. And if we look at the position within the asymmetric unit, we can see that this is, in fact, the case. They're very well positioned to be able to link up. The refinement of the structure turned out to be relatively straightforward, and in the vast majority of the structures that we solve, this is basically how things go. Molecular replacement is relatively simple. We get a solution right away, and it refines easily. The M3 muscarinic receptor represents sort of a contrast. This is one of the most challenging structures we've worked on, particularly at the level of data collection. So these crystals were not only very radiation sensitive and very small, as is typically the case, they also diffracted more weakly than the mu-opioid receptors crystals, as well as most of our other crystals. And because of the very low symmetry, these were actually P1, so there's no symmetry, and they also showed epitaxial twinning. It turned out that data collection involved collecting a huge number of wedges of data and merging them together. In this particular case, as I said, the crystals were epitaxially twinned. They consisted of 2 enantiomorphic lattices which are interwoven with one another. And so we see 2 intersecting diffraction patterns. And this is kind of shown here in one example. This is actually a particularly bad case where the frame has diffraction. It's a little hard to see the spots there, but it has 2 indexing solutions, one of them shown in yellow, the other shown in blue, each of which indexes quite well and which predicts about half of the spots in the frame. So in green are specific positions where the predicted spots overlap exactly. And you can see that there are a number of positions where the spot is reflecting contribution from both of the lattices. And so when we actually try to integrate this data, it doesn't merge well and so on. The solution to this problem in this particular case was actually just a matter of finding crystals that had less of one twin. So we saw that in the crystals that we were collecting, the vast majority of them showed a very overwhelming preference for one or the other twin. They're about equally split between the 2 of them, but in most cases, a single twin was the predominant contributor to scattering on a given frame. And so by collecting a huge number of frames and then discarding any of those that show significant contribution from both twins, and then merging together the ones from the most strongly diffracting wedges, we were able to obtain a complete data set to 3.4 angstrom resolution. In terms of solving the structure then, we choose to do molecular replacement search using the highly homologous M2 muscarinic receptor, which we had solved just prior to this. This was really fortunate because we had just recently solved the structure of the M2 receptor, and it's very homologous. It's homologous both in terms of having very high sequence identity and also in terms of having very high structural homology. And this made molecular replacement quite straightforward. In this asymmetric unit, we had 4 molecules, 4 receptor molecules and 4 T4 lysozyme molecules, which are part of a fusion protein again, and Phaser finds these relatively straightforwardly. Since then, I've gone back and tried to do the search using other search models, other receptors. It turns out that it's much more challenging with certain other search models, although there are a number of them for which this is still quite possible to solve, in particular because the T4 lysozyme is already present and is 100% sequence identity. If you can locate the T4 lysozyme molecules first, it's often possible to find even a suboptimal receptor model and work your way up from there. So in case of the M2 receptor, which is what we actually used to solve the structure at the time, the solution is located almost immediately. Phaser finds it with very high z-scores, and it was clearly a correct solution. As you can see, it's arranged in stacked layers. This is the contents of the asymmetric unit. One thing that was kind of interesting and surprising is that within each asymmetric unit, we actually see 2 layers of membrane and 2 layers of aqueous phase. These are equivalent layers. So within each of these 2 layers, we see different packings, and that's shown here. If you slice through this reconstruction of the crystal lattice, you can see that there's, again, 2 unique packings of lipid-containing layers and 2 unique packings of the aqueous layers. If we rotate this 90 degrees, we can see that these are really quite distinct from one another. The packings 1 and 3 are different, and packings 2 and 4 are even more different. So this contrast between the layers basically defines 4 different types of layers within this crystal, and because P1 is a polar space group, the order in which these layers are stacked within the crystal lattice determines which of the 2 enantiomorphic twins we have. So, for instance, in one case, they're stacked in the order 1, 2, 3, 4 along the positive direction of the c-axis, and in the other case, they're stacked in the opposite order 4, 3, 2, 1. So, just to summarize, the crystals contain 2 different types of receptor packing and 2 different types of lysozyme packing, and the order in which the layers are stacked is different in the 2 enantiomorphic forms, which were interwoven in many of these crystals. In spite of these pathologies, we were able to solve the structure. This shows the electron density from a 2FO-FC map. You can see that it's fairly good. We're able to place most of the side chains with a fair degree of confidence, and we were able to draw some conclusions about the biology of this receptor. So for the final specific example, I'm going to go through the beta-2 adrenergic receptor GS signaling complex. This is the first example of a signaling complex containing a GPCR, and whose structure has been solved, I mean. And basically the idea is that when a GPCR is activated, it binds to an agonist. It undergoes a conformational change, which causes it to open up on the intracellular side. That allows it to interact with the G protein and catalyze nucleotide exchange. So the complex that we were trying to crystallize is the one shown here, the nucleotide-free state, which is sort of an intermediate in this signaling process. To do this required a lot of protein engineering, a lot of biochemistry work. This is work that was done by Søren Rasmussen in our lab. And one of the things that he did was inclusion of an N-terminal T4 lysozyme molecule, which provides hydrophilic surface area on the extracellular portion of the receptor. He also included a nanobody, which is a small antibody fragment, a single Ig domain antibody fragment derived from a llama, which binds at the interface between the RAS domain of the alpha subunit and the beta subunit. Binding at this interface seems to stabilize the complex and facilitate crystallization. So in total, then, the complex that we're trying to solve consists of 6 components: beta-2 adrenergic receptor bound to an agonist, and the heterotrimeric G protein consisting of alpha, beta, and gamma subunits, and then this nanobody, nanobody 35, and T4 lysozyme. So most of these particular subunits have structures available, and the single exception is that the GS alpha subunit is only available in a GTP-bound form. So this model actually is 100% sequence identity, and it turns out, as I'll show in a minute, that it's actually quite different structurally in spite of that. So this is an example where, even though it's the same protein, the large conformational changes ended up complicating the molecular replacement solution. And that's something that we've seen in a number of other cases in our lab as well. So the unit cell parameters are shown here. Based on a Matthews calculation, we anticipate 1 molecule per asymmetric unit. And so to do the molecular replacement search, as others have mentioned, the choice of the order in which we search for the components is really quite important. So to start, we chose to begin with the beta subunit, or the beta-gamma subunit. Beta and gamma are very tightly linked, both structurally and functionally. They never dissociate in the course of normal human physiology. So We search for the combined beta-gamma subunits because we expect that they won't undergo any conformational changes, and we also expect that, and these are also the largest component of the asymmetric unit, largest single component. This was located immediately. This turned out to be very easy to find. We then searched for the receptor, which is similarly fairly easy to locate. We had a very good search model of the receptor in the same conformation in an activated state. So this was also quite easy to locate. And at that point, we got stuck. We weren't able to locate any of the other subunits. And one of the reasons that we speculated might be, one of the factors might be causing this, is that the alpha subunit is composed of two domains, and the nucleotide sits between those two domains. The alpha subunit of the G protein consists of a RAS-like domain, which contains most of the interactions with the nucleotide, and an alpha helical domain. It's previously been shown that these can actually be expressed as separate proteins, and then they interact with each other and recapitulate an intact functional protein. And there's other evidence suggesting that there would potentially be movement of these domains relative to one another. One of those lines of evidence was a series of experiments that were being done in parallel at the University of Michigan by our collaborators, Gerwin Westfield and Yorgos Kiniotis. And what they were doing is they were doing negative stain electron microscopy on the same complex using an antibody which binds to the alpha helical domain. And they observed a huge number of conformations of this complex in the region that is expected to be the alpha helical domain. And this is identified using this antibody fragment. And so this further indicates that there's likely to be conformational heterogeneity in this region. And so in terms of molecular replacement, we then split the alpha subunit into two components, the RAS domain and the alpha helical domain. And although we weren't able to locate the alpha helical domain initially, we were at least able to find the RAS domain, and that was sufficient to allow location of the other subunits, including nanobody 35, T4 lysozyme. And then when everything else is put together, we're able to locate the alpha helical domain. And in retrospect, we can see why this was quite challenging. The alpha helical domain in our structure is positioned over here relative to its position in the inactive, the GTP-bound form shown here. So this is a huge conformational change. It's a 120-degree rotation. The two domains open up completely and expose the nucleotide binding site. And this is consistent then with the electron microscopy data. And this explains why it was so difficult to find the alpha subunit as the intact entity. This is just a summary of the conformational changes going on at the level of the receptor in this complex. We can see that the transmembrane helices undergo a variety of motions. TM6 in particular opens up. And this is basically the sort of changes that we were anticipating the receptor would undergo upon activation. So this structure really has led to some useful insights into how GPCRs transduce that signal across the membrane and how that conformational change at the intracellular side leads to activation of effector proteins such as G proteins. And this is just showing the unit cell, which I think is kind of interesting. The receptors are actually fairly well separated from one another in the crystal lattice. They're separated by a large gap, which is presumably filled with lipid. And then the aqueous layers are much more densely packed, and those contain all of these other subunits. For instance, the T4 lysozyme molecule, the heterotrimeric G protein, and the nanobody. So in summary, molecular replacement is critical in solving GPCR structures. As I said, all of them except for one have been solved by molecular replacement. In most cases, it's actually relatively straightforward. Fusion proteins and chaperones can facilitate molecular replacement. These include T4 lysozyme, can include antibody fragments, and presumably other things as well. And large multi-protein complexes, such as the GS complex, can be solved piece by piece. And that's something that's been kind of a recurring theme, I think, in the last 2 days. We've seen several examples of structures solved using these sorts of techniques. So with that, I'd like to thank the people involved in this work. So Brian Kabilka for his overall leadership of these projects. Ashish Manglik and Seb Grenier led the mu opioid receptor project, and Søren Rasmussen was the leader on the beta-2 GS complex. The M3 project was a collaboration with Jürgen West and Jianxin Hu at the NIH, and Roger Sunahar and Brian DeVries were collaborators on the GS complex, and also Bill Weiss is our major crystallography collaborator in the lab. He provides a lot of oversight and useful advice throughout the process of solving our structures. And I'd also like to thank the National Science Foundation for financial support. With that, I can take any questions.
Questions for Andrew?
I was wondering, missing one or two of those domains after having placed so many other pieces of the puzzle. what did those electron density maps look like?
So the very last piece that we placed was the alpha helical domain, and the electron density there is really quite bad. It's actually, it's resolved, but it's really remarkable in the structure just how heterogeneous the electron density is. And I think the reason for that is just that the alpha helical domain is basically not interacting with much in the crystal. It's pretty much just tethered through the linkage to the other domain, and I think it's just kind of flopping around and occupying whatever space is available to it.
But in principle, that's a small percentage of the scattering mass in the asymmetric unit.
Yeah.
And still you couldn't visualize it after having placed so much other scattering mass.
I mean, when everything else is placed, we can visualize it and we can locate it. I think it's just the high B factor that made it difficult to locate it up until the very end. And the same is true of the T4 lysozyme component in this case.
Hello, Elspeth Garman. If I understood you correctly, you extract your crystals from the cubic phase before you irradiate them. Is that right?
So we usually, it depends on the particular crystal. So we work with two different types of lipid phases. There's the cubic phase and then there's a sponge phase. And the sponge phase is more liquid. The cubic phase is more like, it's got about the consistency of toothpaste. So when we're working with cubic phase, at least I haven't found any good way to actually get the crystals out of it. We typically scoop, we typically scoop the entire thing, and we use a microfocus beam and rastering to actually shoot individual crystals within a loop containing something like 10 or 20 crystals. For sponge phase, it's sufficiently liquid that we can sometimes extract individual crystals. I do know that some people have tried doing things like adding enzymes to degrade the lipids and things like that. We've never actually done any of that. Usually just scooping the whole thing is actually sufficient.
If you ever find a good way to do it, could you let me know?
I would love to have a good way to do it.
Poking around with acupuncture needles, trying to get them out. And it goes very hard, very quickly. It dehydrates the cubic phase, and it's really difficult to extract them.
Yeah, we usually use a humidifier during harvesting. We use a very small room with a humidifier, and that it still happens, but it slows down the evaporation at least.
Any more? No? Okay, well, I'd like to thank Andrew and all the speakers today, I think, for some really excellent talks. And I think Pietro is going to provide some closing remarks.
Can you hear us? Yes. All right. What time is it? Yes, it's time to go home, and we will keep it short. I'm not sure how sweet. We are very pleased with how it went. We hope to have feedback from all of you, especially from the young people in the audience, I guess. We got together earlier to discuss this, and we thought we would just throw a couple of comments out there. One was that it doesn't seem that the software development community can quit working on molecular replacement just yet. We're not sure if it's because of need or because of love of the subject, but since the last molecular replacement meeting, a lot of things seem to have happened, and we are looking forward to another MR meeting in 6 years or so from now. I think we are. At some point, perhaps it will have to be dropped, but it doesn't seem the case that it can be dropped yet. And then whoever has confronted structure solution by MR very often feels that this is kind of unique problem, perhaps because we think heavy atoms, there's only so many heavy atoms, and in the end we have to solve a Patterson, while proteins are a bit wider than the choice of heavy atoms. And so there is certainly a stage of the procedure where you are confronting this idea, I'm alone in this world and nothing can help me unless things go straightforwardly. But on the other hand, I think one of the themes that was coming up again and again is that if you read the literature, especially the proceedings of this meeting, when they're out, you will find some guidance and you will get some ideas. Perhaps you'll develop something which is unique to your problem, but more often than not, that's the advantage of the young people. They are standing on the shoulders of giants. Then, one particular interesting
topic seems to be where are we going with finding models, and why is it that lately we seem, or not so lately, perhaps for quite a while, we have been depositing in the PDB structures that have a fold which we've seen before or belong to a family that we've seen before. And here perhaps we want to start thinking of the context in which we are operating, which is inevitably it's human endeavor, it's a socio-economic context. So which targets do we choose? Do we choose them on the basis of how helpful they are or how easy they are? And it's quite a complex set of questions, and I think it was good to be reminded of them. And then perhaps about the complexity of ways you have to generate your model, complexities of ways you have to finding the placement of your model, and complexity of ways you have to refine your model to the extent that you get to your structure solution. I think this meeting was quite good in that way, and we hope that you go away with a sense of complexity. And then we start some thanking, because obviously it's been quite a collective endeavor. CCP4 at large, first of all, and the funding, but in particular the working groups who are steering it, Working Group 1 and Working Group 2, who were very instrumental in guiding us, discussing with us the choice of speakers, topics. We couldn't have done it without them. We should thank the organizers here locally, because I think it was quite a great venue and there was a lot of work done to prepare for this, and in particular we would like to thank Shirley for all the organizing that she's offered us towards this. Is Shirley here? Because okay, perhaps she's dealing with organization. We had some flowers for her. Then, and Damien and Laura, who are also probably busy and are not here. We want to thank Early McCoy, who was effectively the third member of this small group. And of course I thank Helen, although we had some, and she thanks me. Is Early there? Where's Early? She's not there. All right, there are some flowers here if anyone wants, but we can give Helen's hers. And then, well, I want to thank shortly my friend and colleague Luigi De Colibus in Oxford, with whom I started brainstorming this even before I met Helen, and I think some of the ideas I brought when I first met with Helen came from him, because he reads the literature. And then we want to thank the speakers, because some of them came from very far away and it's the holiday season. Some of them came from not that far away, but they may have encountered some difficulties in getting here, and it was a great privilege to have them all, and I think they did a brilliant job, and so I think we should join in an applause for them. And finally, Helen and I want to thank you all, because you were not terribly proactive with your questions, but we trust that you had good questions in your heads, and otherwise socially it's been very good. We hope you met some colleagues and made friends and can keep contributing to the sense that this is actually quite rare in some ways as a scientific community, because it's unites rigor with passion, perhaps, or in any case with generosity in sharing results, ideas, and so thanks to all of us. And I think that's it. Yes, I can take care of it.